Autoimmune conditions such as type 1 diabetes, rheumatoid arthritis, celiac disease, multiple sclerosis, and lupus affect 8% of Americans. Now we know a lot about these conditions and even the genes that cause them, but that wasn't always the case. Early 20th century observations noted that skin grafts between individuals were often rejected, suggesting some form of genetic incompatibility. John Dosset and Peter Gorer independently observed leukocyte white blood cell antigen variations in mice and humans, laying the groundwork for HLA discovery. In 1965, Jean Dosset, along with Jean van Rood, established the first standardized system for naming human leukocyte antigens, a crucial step for further research. The 1960s saw the formation of international workshops to share data and refine HLA nomenclature. The term quote-unquote human leukocyte antigen, HLA, was officially adopted in 1968. Researchers identified different classes of HLA molecules, with HLA-A and HLA-B being the first ones discovered. This provided a more nuanced understanding of the HLA system. There are a couple of different strategies that these early genetic researchers could use to actually identify and map the HLA genes to where we currently know they are located. All of these techniques are, to put it lightly, inefficient. Here's the strategies that these early researchers employed. One of them is somatic cell hybridization, the crux of which was essentially to fuse the cells of two different samples of different species or individuals, and whatever is retained in the hybrid could help the researchers locate certain genes. The second strategy they often employed is karyotyping, which is essentially the study of morphology of the chromosomes and can be helpful in determining various deletions or insertions, and through that can be useful in identifying the location of a certain gene. So, for an example, if somebody has an autoimmune condition, it would be perhaps useful to conduct karyotyping on their genome to find if they have any deletions somewhere, and if they do have deletions somewhere, perhaps that deletion is exactly in the region that is relevant to the HLA gene. Or maybe it isn't at all, and that's why when you don't have solid data like this, and you have to do sort of guessing, shot in the dark, you have to do a lot of work, and you have to gather a lot of statistical data. Nonetheless, eventually the HLA gene cluster was located, and now you can even find it in your raw DNA file that you got from MyHeritage or Ancestry or 23andMe. Isn't science awesome? HLA genes all essentially fall right on their centromere of their 6th chromosome. HLA-A falls on the short arm of the 6th chromosome, right by the centromere, whereas the rest of the HLA genes, including HLA-B, HLA-C, HLA-DR, HLA-DQ, and HLA-DP, fall on the long arm of the 6th chromosome, also right by the centromere. HLA-A is responsible for a lot of things. Among them is white blood cell count, granulocyte count, hyperthyroidism, hemoglobin levels, and multiple sclerosis, although the odds ratio for that is quite close to 1. Overall, this gene isn't that involved in various autoimmune conditions. By the way, fun fact, in this HLA variations implicated in white blood cell count, the allele that causes decreased white blood cell count peaks in South Asians at 37% frequency and is lowest in Koreans at only 1.6% frequency. HLA-B is implicated in lymphocyte count, A2M RAP, which is a very important unit to measure in the context of autoimmune disease, as A2M is a protein involved in clearing enzymes and other molecules from the bloodstream. Psoriasis, primary sclerosing cholangitis, monocyte count, as well as various allergies. HLA-C is directly implicated in immune function, as can be measured by the ratio of GZMA to KIR-TDL3 protein levels. GZMA, produced by cytotoxic T lymphocytes and natural killer cells, is a protein that induces programmed cell death in target cells. KIR, an inhibitory receptor on NK cells, can mitigate the cytotoxic activity of these cells, potentially affecting GZMA-mediated cell death. HLA-C also has a great impact on lymphocyte count, white blood cell count, neutrophil count, killer cell immunoglobulin like receptor DS2 levels, ultralight colitis, psoriatic arthritis, hypothyroidism, and interestingly, HLA-C is not implicated in allergies unlike HLA-B, neither is it implicated in multiple sclerosis. HLA-DR and DQ is the most well-studied part of the HLA region. This part is usually well sequenced and covered in every commercial DNA test, including MyHeritage, Ancestry DNA, and 23andMe. It is implicated in rheumatoid arthritis, hypothyroidism, eosinophil counts, multiple sclerosis, asthma, Vogt-Koyanagi-Harada syndrome, Addison's disease, bullous pemphigoid, vitiligo, leprosy, type 1 diabetes, and various allergies. HLA/DP is implicated in mostly the same things that HLA-DR and DQ are implicated in, but it has been studied in depth for its connection with bone mineral density, hypothyroidism, hepatitis B, and Graves' disease. Thank you for watching my video. I think it is relevant to mention that in the description of this video you will find the link to purchase my trait predictor, which is a tool I developed that allows you to generate a health and traits report for yourself with a raw DNA file from MyHeritage, 23andMe, or Ancestry. It includes a very thorough health and traits panel and even an HLA gene panel. Thanks for watching. Goodbye.